Okay, yeah, it's recording now. And then that is the information about the crossing over, and we spoke about what crossing over results in. So it results in exchange, in exchange of genetic material. It also leads to genetic variation amongst the gametes and the offsprings. So that's the reason that you look different to your siblings, and that's the reason that you and your siblings do not have the same genetic material. Then we looked at metaphase 1. In metaphase 1, we said that the chromosomes align themselves in pairs at the middle of the equator. And I said the word pairs is very important because, as you can see, if you look at the red ones, they are 2 chromosomes that are attached to the same spindle fiber. So they are a pair. And this is just after crossing over. We also explained that these chromosome pairs align themselves randomly in the middle of the equator. There is no particular order that they follow. So, and that is stated by the law of independent arrangement. So that is metaphase 1, and that's all the information showing you the independent arrangement. Then in anaphase 1, here we speak about the whole chromosome being pulled to opposite poles. So what will happen first here is that the spindle fibers are going to contract, and when they contract, they separate the homologous chromosome pairs, and then only a whole chromosome will be pulled up, and another whole chromosome of the same pair will be pulled down. That's exactly what we have in anaphase 1. Now it's the terminology. What is being pulled here? Whole chromosome. You can't just say a homologous chromosome pair is being pulled, because that is not true. You can't say a chromatid is being pulled to opposite poles, because that is also not true. Please also make notes. I think I did mention it last week. If they say opposite poles, that's what they want, opposite poles. If you say opposite sides of the cell, you are not going to get a mark for that. Okay, then that was the information for anaphase. Then in telophase 1, so you will have the whole chromosome reaching the opposite pole. Then the spindle fibers will disintegrate or will disappear, because remember their function was to make sure that the chromosomes reach the opposite poles. Now that the chromosomes have reached opposite poles, they start to disappear. Then the cell membrane will constrict in preparation for cytokinesis. That is the division of the cell. Cyto means cell. Kinesis means division. So the division of the cell will start so that we end up having 2 cells at the end of telophase 1. Right? Now, that's not the end. Now, the cells, both of them, are going to undergo meiotic division too. Remember, this is still a recap, guys, so don't mind my slides moving very fast. That is still a recap of what we've covered. Then in prophase 2, those 2 cells are going to continue. Same thing that we spoke about. Nuclear membrane disappears, nucleolus disappears, spindle fibers start to form. And then here, you can see that the chromosomes are not in pairs. We started with 6 chromosomes, which were 3 pairs in prophase 1. And in prophase 2, in each cell, we are getting 3 chromosomes. They are still replicated because they have both the chromatids joined by a centromere. So that's what happens. There is no crossing over in prophase 2. Then in metaphase 2, the chromosomes arrange themselves randomly, but now these are single chromosomes. In metaphase 1, we had pair of chromosomes arranging. Now in metaphase 2, single chromosomes arranging. In anaphase 2, spindle fibers will contract and they are going to split the centromere, leading to chromatids or unreplicated chromosomes. Even if you just say chromosome, unreplicated chromosomes being pulled towards opposite poles. Remember, in anaphase 1, it was the whole chromosome that got pulled to opposite poles. Now, this is the end of telophase 2. So yes, you're seeing 2 cells here at the end of telophase 2, but there's actually 4, because we just used an example of 1 cell from the 2 cells. So at the end, it will be 4 cells. This is what we have. And I like this diagram because you are able to see in each phase what happens in prophase for each division, right? So prophase 1 compared to prophase 2, metaphase 1 compared to metaphase 2. Let me figure out how I can write on the slides. It's not doing what I want to do. So I might have to go back. Can, right? Okay. It's becoming way too slow. I knew that PowerPoint will probably slow down everything. So just give me a second. I'm going to go back to opening a PDF document, which has a smaller size. Okay, just a few seconds. I'm almost there, just a few seconds. Okay. She said we don't have the slide on these notes. Okay, that's fine. All right. Let me check if it's sharing. Let me go back here. Okay, it was not sharing. Okay, 5 seconds. The slides should be back on, but now I'm just using PDF because that's better. Okay, so now you should have the slides, and we speak about the importance of meiosis. This is important for you to know also. They do have a way of asking you. One, the importance of meiosis is to produce haploid gametes. Remember the word haploid? We covered the words haploid and diploid last week, right? So haploid means half the number of chromosomes. And when we speak of a gamete, we are speaking of sex cells, meaning sperm cells in males and egg cells in females. So these processes, they actually do take place in us human beings to produce the egg cells, and then in males to produce the sperm cells. Also, the importance of meiosis is to half the chromosome number, because we are moving from a diploid to a haploid. So if we speak about a human cell has 46 chromosomes. It will then undergo meiotic division 1 and then meiotic division 2. Now, at the end of meiotic division 2, each cell will have half the number of chromosomes. That means in this case, each cell will have 23 chromosomes. Sperm cells have 23 chromosomes, egg cells have 23 chromosomes. Okay? Then also, the importance of meiosis is that it introduces genetic variation, genetic difference. That is why we have different genetic material as human beings. So, and that is introduced through crossing over. The reason there is genetic variation is because of crossing over, and it's also because of how the chromosomes randomly arrange themselves in the middle of the equator during metaphase 1 and metaphase 2. Then, sources of genetic variation, they do also ask this. So the reason there is genetic variation is because of crossing over, it's because of independent arrangement of the chromosomes, it's because of random mating, partner selection is random at all times, and also random fertilization. From the millions of sperm cells that are released during ejaculation, there is no particular order of which sperm cell is going to fertilize which egg cell. So that is random fertilization. From the millions, we just need one to fertilize an egg cell. So that is also a source of genetic variation. Mutations, so there's certain changes or mistakes that can happen during meiosis that will result in the variation of gametes. Remember, we also spoke about mutations in DNA. That was gene mutation. So in meiosis, we speak about chromosomal mutation, which I will cover in this lesson. So these are just diagrams that you can look at in your own spare time. Now, these are the comparisons between meiosis 1 and meiosis 2, and I think we covered them in detail. I'm not going to go through them now. This is what we need to cover. And they will definitely assess this. And now I think in the past 3 years, it's been more of an application question. So you need to understand the theory first and then follow whatever example that you are given in a test and answer accordingly. So what we've been covering is normal meiosis. Everything that we've mentioned in each phase would have been how meiosis needs to occur normally. Now, what we are going to cover here is abnormal meiosis. Some of the things that occur during meiosis that are abnormal, certain changes or mistakes that may occur. So that's what you need to remember. What was normal meiosis? We had 2 divisions, and everything that we've mentioned in prophase 1, metaphase 1, anaphase 1, telophase 1, prophase 2, metaphase 2, anaphase 2, telophase 2. That was normal. Now, we will speak about certain things that may not happen. And the main phase that we are going to focus on is anaphase 1 and anaphase 2. Now, a type of abnormal meiosis that we cover is called non-disjunction. Non-disjunction is the failure of chromosomes to separate during meiosis. Remember, when the chromosomes separate, they actually separate in the anaphase. So that will be anaphase 1 and anaphase 2. If we remind ourselves, in anaphase 1, we are expecting the homologous pairs to be separated. This is what we are expecting in anaphase 1. We want the pairs to be separated, and then the whole chromosome moves to opposite poles. So this whole chromosome will move up, and this whole chromosome will move down. That is normal meiosis. Then in anaphase 2, we are expecting the centromere to split, and only a single-stranded, they'll never take that, not single-stranded, only an unreplicated chromosome or a chromatid gets pulled to opposite poles. So remember, this is anaphase 1 and this is anaphase 2. That is what is supposed to happen. Now, when it comes to abnormal meiosis, it is possible that this pair of chromosome that is supposed to separate for a whole chromosome to be pulled to opposite pole fails to separate. Now, instead, let me get my pens. Instead of each cell having the chromosome, let me get my other red color. Okay. Trying to show you here. Okay. So this is our pair of homologous chromosomes in the middle of the equator, that is metaphase 1. So my black pens represent one replicated chromosome, my blue pens represent the second replicated chromosome, they are homologous pair. Now what we expect in anaphase 1, we're expecting this whole chromosome to move up and this whole chromosome to move down. But it is possible that during anaphase 1, there's a failure of the chromosomes separating. So what ends up happening? The whole pair moves up, or the whole pair moves down instead of being separated and one chromosome up, one chromosome down. That is non-disjunction occurring in anaphase 1. So what do we have here? There is the information. Homologous chromosome pair does not separate. Homologous chromosome pair does not separate, and both homologous chromosomes get pulled to one pole. But now what happens in the other pole? Remember, these were a pair, and one was supposed to go up and the other was supposed to go down. But if both of them go up, that means the other side of the cell does not receive the chromosome pair, does not receive that chromosome from the pair. Now that means another cell will be one chromosome short. Now that is non-disjunction occurring in anaphase 1. It is possible for it to occur in anaphase 2. So remember, in metaphase 2, we will have chromosomes arranging themselves singly in the middle of the equator. So these are my chromatids joined by a centromere. Now, what is supposed to occur in normal meiosis? The centromere is supposed to split so that the chromatids are pulled to opposite poles, or we can even say the chromosomes are pulled to opposite poles. But in abnormal meiosis, it is possible that during anaphase 2, now instead of a chromatid going up and a chromatid going down, the whole chromosome, if the centromere does not split, the whole chromosome moves up, and then the other side of the cell doesn't receive half of that chromosome or doesn't receive the other chromatid of that chromosome. That is non-disjunction in anaphase 2. What has been the difference here? In anaphase 1, it will be the whole pair not separating. Then in anaphase 2, it will be the whole chromosome not being split, meaning the centromere is not split, and then the chromosome only moves to one side of the pole. The other pole does not receive a chromosome of that pair. That is what is abnormal meiosis. Let's look at it in detail. So non-disjunction in anaphase 1 or non-disjunction in anaphase 2. Please note, non-disjunction occurs only in these phases, not in prophase, not in metaphase, and not in telophase. So what do we end up having here? So if our cell were to have 2 chromosomes, this is the parent cell, right? And has 2 chromosomes. Now, what are we expecting during anaphase 1? We're expecting these 2 chromosomes to be separated. These 2 chromosomes to be separated so that one cell can have this one chromosome and the other cell can have the other chromosome. But what has happened here? Both chromosomes have now been moved to one side of the pole, resulting in a cell having an extra chromosome. This is what we are supposed to have here, 2 chromosomes. At the end of meiosis 1, we're supposed to have 1, 1, and 1, 1, 1, 1. This is what we are supposed to have if we have meiosis occurring in a cell that has 2 chromosomes. Remember, we're going to have the chromosome number. But what do we end up having? We are ending up with a cell at the end of meiosis 1 having an extra chromosome. What about the other cell having no chromosome? So that means from the gametes that are formed now, we are having 2 cells with 2 chromosomes. And what about the other gametes? They don't have chromosomes. So that is non-disjunction. These are the mistakes that can occur. Now, this is mutation. We speak about certain conditions and syndromes that we find in kids due to some of these mutation or mistakes that occur during meiosis. Now, let's look at what happens when there is non-disjunction in anaphase 2. We started with 2 chromosomes, we are happy, and then at the end of meiotic division, yes, we have the number of chromosomes, we separated the homologous pair. 1 chromosome in the cell. There was no mistake in meiotic division 1. Now, the change occurs in anaphase 2, meiotic division 2. What has happened? We are expecting this chromosome to be separated and this chromosome to be separated because the centromere is going to split, right? So that we can have this cell having 1 chromosome and this cell having 1 chromosome. This cell having 1 chromosome and this cell having 1 chromosome. But what has happened now? That means in this particular cell, the centromere did not split. Now, both chromatids of the same chromosome move to one side of the pole, ending up with 2 chromosomes in the cell. So this cell ends up having 2 chromosomes. This one, 0 number of chromosomes. This one, 1 and 1. That means there was non-disjunction there. This is abnormal meiosis. Let's look at it again. Non-disjunction in anaphase 1. Remember to stop me any second and ask a question. Non-disjunction in meiosis 1. What did we have in metaphase? So remember, you are seeing a green chromosome and another green chromosome, a purple and another purple. So these 2 were a pair and these 2 were a pair. But on this side, the chromosome successfully separated, the pair successfully separated. The green chromosome goes up, and then the purple one goes down. But what happened here? The chromosomes did not separate. So that means this one, this pair ends up going up. So what is this leading to? This leads to a cell having one extra chromosome. There, instead of having two, it's supposed to have only two chromosomes. What does it have? It has 3 chromosomes, right? And then what happens here? From the 3 chromosomes now, they are going to undergo meiosis II. They will continue with that same mistake. What is the result? This cell has an extra chromosome. This one has an extra chromosome. This one has 1 and 1. Whereas it's supposed to be 2, 2, 2, 2, because the chromosome number needs to be the same. Let's look at non-disjunction in meiosis II. Now, we are happy with meiosis I. Separated properly, separated properly. But now let's see what happens here now. Instead of this particular chromosome, the centromere is supposed to split so that we can get a chromosome here. You can see we are happy with this one. There was good separation. But here, this one did not separate. So what does this lead to? It leads to us having this cell having an extra chromosome. It's supposed to have 2, but it has 3. We're going to count them as 1, 2, 3. Okay, and then this one, what does it have? It has 1 chromosome. So it's supposed to get 2, 2, 2, 2. But we're no longer having 2. We're having 2 plus 1, meaning is 3. And here we're having 2 minus 1. N is the supposed haploid number. That's why you're having N minus 1. So we're supposed to have 2, but 1 is not there. So it's minus 1, meaning we're only having 1 chromosome. Now that is what is happening. So if we're to bring it back
Hi, ma'am.
to
I have a
yes,
question, ma'am.
yes,
Hi, ma'am. I just wanted to quickly ask, will we ever get an instance where, let's say, the non-disconjunction happens in both anaphase 1 and anaphase like 2?
we get that question a lot from the classrooms. I feel like anything is possible with the question papers. So I would say that is highly possible. We don't know which year they're going to bring that question, but so far it's been just a non-disjunction occurring in only one division. But I feel like it is possible for them to give us a question like that, where they say non-disjunction occurred in anaphase 1 and continued to also occur in anaphase 2. Now, this is when application comes in. You will need to read into the diagram and you check the chromosome number. How was it affected in anaphase 1 and also how was it affected in anaphase 2? I wouldn't say they'll never give you that question. I think it is highly possible that they can give you. All right. It was Lilo, right?
Yes, thank you, ma'am.
Okay, thank you so much. All right. Let's think about this if it were to happen in human beings, right? If we are looking at cells for human beings, remember in the chromosomes, we are having 46 chromosomes in our human cells. What do we expect? We are expecting at the end of meiosis 1 to have 2 cells which have 23 chromosomes each, and those 2 cells to undergo meiotic division 2 and also have 23 haploid chromosomes. Now, this is us being happy. But if chromosomes fail to separate either in non-disjunction anaphase 1 or non-disjunction anaphase 2. So if it does happen this side, what do we end up having? One cell will have 1 extra chromosome and the other cell will have 1 less chromosome. So what am I talking about? So if it's the cell that has 1 less chromosome, that means this one will get 23 chromosomes. Then another one will have 22 chromosomes, and then another one will have an extra chromosome. This one has 24. This is abnormal. This is abnormal because if this is a sperm cell, so now this will be a sperm cell with 22 chromosomes, and then if it fuses with a normal egg cell, which brings 23 chromosomes. So if you add these chromosomes together, you are actually getting 45 chromosomes. That is abnormal meiosis, and the person will probably have a syndrome. If they do become alive, they will probably have a syndrome because a normal person is supposed to have 45. Wow, 46. So the 45 would be due to mutation. But now what happens if this is the sperm cell that fuses with an egg cell that is normal? So we'll get 24 chromosomes from the sperm cell plus the normal number of chromosomes from the egg cell, which is 23. This brings us to 47 chromosomes. Now, this is mutation and it leads to what we call Down syndrome because this person now has an extra chromosome. Yes, these people are alive, but now they have the syndrome, which is Down syndrome. So let's get to it. So this is application, guys. You read into the question. I think we will do a few questions. You need to read into the question and then you find out the story. Where did non-disjunction occur? Was it meiosis 1 or meiosis 2? And what did it result in? If they're speaking about human beings, if they're speaking about a certain animal that has 8 chromosomes, you figure that out. So if they speak about 8 chromosomes, what is the expectation? We are expecting each cell to have 4 chromosomes. Right? But now if they say there was non-disjunction that happened or wherever, now you can keep track. Okay, that means a cell might have 1 extra and another cell might have 1 less. So the one that will have 1 less, that will be 3 chromosomes. The one that will have 1 extra, that will be 5 chromosomes. But what is our normal number that we're expecting? 4. So you read into the scenario. That is very important. Now, with Down syndrome, there isn't a lot of things that you need to learn here. You just need to know the name, which is Down syndrome. And this is due to mutation or non-disjunction. And this is non-disjunction that normally happens in chromosome pair number 21. So we are supposed to have only 2 chromosomes in that pair, in pair number 21, chromosome 1 and chromosome 2. But due to non-disjunction, we end up having the 3rd chromosome. So Down syndrome is also referred to as trisomy 21. Tri means 3, 21 is referring to the pair. In chromosome pair number 21, there are 3 chromosomes that results in what we call Down syndrome. Now, the chance of non-disjunction occurring in sex cells, it does increase with age. So that is why they normally encourage females to have babies by a certain age, because non-disjunction has a higher chance of occurring in females most of the time, around, I think, more than 40 years or 38. I'm not sure about the age, but I think in recent times we've seen females giving birth to healthy kids at the age of 40 or 50 or whatever age. Okay, so what are we taking here? Trisomy 21, Down syndrome. This is due to non-disjunction occurring at chromosome pair number 21. This is how people with Down syndrome normally look. They will have a small nose. They normally have, I think, mental situations there. Their muscle tone becomes different. There's normally challenges with heart defects, hearing loss, and all of that. But they don't ask you about the symptoms most of the time. So I wouldn't say we stress about this. This slide is super important. Super, super, super important. They ask you about non-disjunction of chromosome pair number 21, that is Down syndrome. So they ask you, how does Down syndrome, or how does non-disjunction lead to Down syndrome? What do you say? Non-disjunction of chromosomes at pair number 21 or at position 21. During where it's either anaphase 1 or anaphase 2. We do not know in which phase, whether it could be 1 or 2. So you mention it unless otherwise stated in the scenario. Now, this forms abnormal gametes with an extra copy of chromosome 21. Extra copy meaning 1 more chromosome. Now, the fusion between the normal gamete and an abnormal gamete. So normal gamete having 23 chromosomes and abnormal gamete 24, meaning 1 extra chromosome. That leads to Down syndrome because this person will end up having 47. We add this and that, and the person ends up having 47 chromosomes. Okay. I think how a zygote is formed in trisomy 21, that's exactly what would have happened. So what do we have here? So 22, 22, 22, 22. That means non-disjunction occurred in meiosis 1 here, because how can we see that? So this cell is supposed to have 23, and this one is supposed to have 23. And at this point, there is one extra this side, we're having 24, and one less this side, there is 22. Now, we are able to see in which division or in which anaphase non-disjunction occurred. Then what do we end up having here? So that means all the cells actually have an abnormal number of chromosomes, because all of these cells are supposed to get 23 chromosomes each. But what do we have? One short, one short, one short, and one extra. Now, if this is an ovum, this ovum brings in 24 chromosomes, and this sperm cell brings in 23 chromosomes, we end up having 47 chromosomes in the zygote. That is trisomy 21. Let's familiarize ourselves with this particular diagram because it is very important. Who can remind me? Oh, it's on my screen. I was about to say, what do we call this particular diagram? That is a karyotype, right? That is a karyotype. But who can tell me, is this karyotype, please do unmute and tell me, is it of a male or a female? The karyotype, anyone? Is it a male karyotype or a female karyotype?
Male.
Male, definitely male, because we can see here we are having XY chromosomes in the chromosomes. Then there is 23 pairs here. Now you need to read into this. They can ask you, is there a syndrome that you are able to see from the diagram? Yes or no? Yes. Please give us the name of that particular syndrome. Down syndrome. Then they ask you, why are you saying it's Down syndrome? What are you going to reference? What is happening here? There is an extra pair of chromosome in chromosome pair number 21. Then they may ask you, are these chromosomes replicated or unreplicated? We can see these are unreplicated chromosomes. So from pair number 1, this is chromosome number 1, chromosome number 2, and they are unreplicated. They will replicate during DNA replication. Now, anything else that is on the rest of the slides, it's not really that important, something like this one, but this is very important in terms of application. I'm not going to read what it's saying here, but it's basically telling you to be able to read into the question and then apply theory. Let's work together. Let me see the diagram below. How many minutes am I going to give you to do this one? Fun fact, this question is from last year, November 2025. Please do answer this. What do you need me to project? I'm not sure if you have this activity on your slides, maybe not, because this was recently updated. But what do you need me to project? Do I need to project the diagram or try and project both, maybe? Try and do this. Is that better? Please do just unmute and communicate so that I know what you are able to see on your side. Yes? Is this fine? Thumbs up, yes, no? Hello, guys. Okay, thumbs up. All right. I'm going to give you 6 minutes to answer this. 6 minutes to answer this. If it's better for me to just send a screenshot on the group, please do let me know so that you are able to see what is happening here. So I need you to answer all the questions. This is from November 2025. That is the most freshest question paper. So let's answer. This was not 100% easy, especially the last question, but let me be quiet. So 5 minutes, it's 1943. At 48, we are answering. Let me know when you need me to scroll down, or if it's better if I just take a picture and send to the group. Please do let me know what works.
Just send us a screenshot on the group.
Okay, let me do that. Time flies, guys. 3 minutes. Don't kill me. Okay, group A. I'm just trying to take the pictures and send. There you go. Please do try your best. Please, please, please. I'll give you 1 more minute. All right, I think we can mark. Can we mark, guys, or you need maybe 2 more minutes? Please do indicate something. If you want us to mark, you're ready on your side, thumbs up. If you need extra minutes, just unmute and do say that you need extra minutes so that I know.
Ma'am, can we please have 2 more minutes?
Perfect. Okay, I think we can start now. Lilo, you'll be the first one to give us answers. Okay, let's mark quickly, guys. Identify structure A. So what did you say?
I said the structure A is our centromere.
Centromere. Centromere, that is correct. Okay, then our second question will be answered by Nombuso. Give the biological term for the sex chromosomes. What did you say, Nombuso? Sinako, what did you say? For 2.1.2, we are looking for the biological term for the sex chromosomes. Okay. Yes? Anyone? Anyone, please do give me an answer.
Is it not 2 pairs of homochromosomes?
The biological term for the sex chromosomes. What is the biological term for that sex chromosomes? The XX or XY?
Is it not female? Is it not a female?
Female? No, it's not. Someone is saying something? Octosome. Autosome. No, it's not autosome. We're not moving, guys. You need to give me the correct answer for this one.
Ma'am, is it not gonosomes?
Gonosomes. Gonosomes. Okay, we said this is the central gonosomes, guys. Thank you. For 1 mark, describe how these chromosomes would differ in the somatic cells of a male. So which ones are they talking about? They're speaking about these ones, right? They're saying the diagram below represents the sex chromosomes in a female and they've labeled them XX. Now they want you to describe how these chromosomes will differ in a somatic cell of a male. Cindy, so what did you say? Okay, anyone? If you're ready to answer, I'm not sure which one works. Raise your hand, or I'm calling out people's names. There are no responses. So please do raise a hand. At least I can see who's ready to answer on my side, because I'm calling out names and there are no answers. Iviwe, what did you write? Okay. Anyone, please do give me an answer. It can be anyone, guys. How these chromosomes differ in the somatic cells of a male. Okay, we're not going to move. We'll wait for an answer. We're not moving up until we get answers.
Yeah.
I can't hear.
Okay.
You're cutting, Mulalo. I can't hear you.
Hello.
Yay.
Yes, I think is the network thing. So okay, in male the default will be XY.
That is correct. The difference will be in males, you are going to have XY. So remember, in females, you have XX, and in males, you have XY chromosomes. That's a mark. Then an exchange of genetic material takes place at region P. There is region P they have shown you in the diagram. Name the process above. I don't want the same people answering, guys. We are all in a lesson, please. So when I call out your name, please do answer. Yes, I'm listening. Crossing over. That is correct. Thank you. Crossing over. Please note, if you wrote cross over, no mark for it. It has to be crossing the process, crossing over. The mark is at the end here.
I said it's not for me.
What did you say? Please repeat your answer. I didn't hear that. What did you say? Was it Zania speaking? Okay. So what I was saying is the correct answer is crossing over. So if you wrote crossing over, tick. But if you wrote cross over, just the word cross, and then you wrote over, this is wrong. They're not going to mark you for this. Okay. Then 214B, identify the phase during which the process named in question 2.1, 2.1.4 takes place. Which phase is it?
Phase one.
It,
Phase one.
Phase.
it's Siasma.
Phase what, Miss?
Prophase.
Prophase
Prophase
1. If you wrote just prophase,
It's gonna
you
go,
didn't.
ma'am.
Which one?
Then go.
Okay, so we are at 2.1.4. When they ask for a phase, we are here at B. The correct phase is prophase 1. I'm hearing an answer about gonosomes. I'm not sure which question that answer is for. So here they've asked for a phase. It has to be prophase 1. If you don't have 1 next to your answer, wrong. So the tick is just after 1. Okay? Then C, we are busy with 2.1.4C. Give one reason why the process named in question 2.1.4A is important. Give one reason why crossing over is important. What did you guys say?
Because of exchange of
Because of
genetic
the exchange of material. And
Okay. Yes, I'm listening. There was an end.
Genetic material.
because of
Okay,
the
when
exchange of
this
the
material is exchanged,
material.
what does it lead to? So the exchange of the genetic material is what happens, yes. But give one reason why that exchange is important. The correct answer is to introduce genetic variation. So the material becomes exchanged so that there is variation in the gametes. That is the correct answer. To introduce genetic variation in gametes. Because remember, this process occurs so that we produce gametes that are not genetically identical. To introduce genetic variation in gametes. Okay, then 2.1.5. Explain how the structure of the chromosomes will differ at the end of meiosis 2. Okay, please give me an answer for 2 marks. What are they asking for here? Yes, I'm listening.
Explain.
Ma'am, I said the centromere will split as the chromatids move to the opposite poles of the cells. Therefore, 4 cells are formed at the end of the division.
Okay. Now that is what happens in meiosis. Let's look at what the question is specifically asking for. So you're speaking about the separation of chromosomes, and that will have 4 cells at the end of meiosis 2. Correct. But is it answering our question? No. They want you to explain the structure of chromosomes. How would it differ at the end of meiosis 2? Let's see what we are given here. We are given this particular structure of chromosomes, and these chromosomes are replicated and they are in pairs. So at the end of meiosis, how is the structure of chromosomes? Remember, the chromosomes are unreplicated. That's what they're looking for. At the end of meiosis 2, you can speak about the chromosomes having 1 chromatid. That is the structure of chromosomes. That's what you are answering. So the 4 cells correct in terms of what meiosis is and what happens, but it's not specifically answering the question. So the answer here is chromosomes are going to be different due to crossing over, consisting of only one chromatid. So remember, in terms of the structure of a chromosome, this is a chromosome made up of 2 chromatids, and this is a chromosome made up of 1 chromatid. At the end, the chromosomes are made up of 1 chromatid in each cell. That's the structure of the chromosome that we are answering. Now, I want to hear the business for 2.1.6. Non-disjunction of these chromosomes occurred during meiosis, and a gamete with an extra chromosome is formed. Non-disjunction of these chromosomes occurred during meiosis. That's what they tell you. And what are they telling you? They are telling you that it's a gamete. A gamete is supposed to have 23 chromosomes, right? And they're saying this gamete with an extra chromosome is formed. So what does this mean? It becomes 23 plus 1. Now, if this particular gamete, which has 24 chromosomes, right, is fertilized by a normal gamete. So if this one with 24 chromosomes is fertilized by a normal gamete, the resulting zygote will not be normal. What do they want you to explain here? Explain the genetic composition of these zygotes. For how many marks? 3 marks. So what are you going to talk about here? You are going to speak about the normal gamete. A normal gamete is supposed to have 23 chromosomes. But what are we speaking about? A gamete. A gamete should either have an X or a Y, because it will be 22 plus X or 22 plus Y. So what are we going to have here? The abnormal gamete will have 2 X chromosomes. Why are we saying that? So instead of being 22 plus X or 22 plus Y, but I'm going to focus on X. Why is that? Because the chromosomes that we are given are XX chromosomes. Instead of a gamete having 22 plus X, it will actually have 22 plus XX. So it ends up having 24, right? But now, if it were to be fertilized by a normal sperm cell, and remember, this is from a female, meaning it's 22XX, and a sperm cell is bringing the Y chromosome or the X chromosomes. So this will be XXX. This is the new X that has been brought in, or we can have the resulting zygote having XXY. What is happening here? This is the 2 X's that were not separated, and this one is from the male. And this also were the 2 X chromosomes that were not separated, and this one is from the male. So what do I have in the memo? The abnormal gamete will have 2 X chromosomes, XX. If fertilization occurs with a sperm cell, remember a sperm cell can carry an X chromosome or can carry a Y chromosome. If a sperm cell carries an X chromosome, that means the offspring is going to be a girl. If the sperm cell carries a Y chromosome, that means the offspring is going to be a boy. The gender of the offspring is determined by the sperm cell. So if fertilization occurs with a sperm cell carrying an X chromosome, the resulting zygote will be XXX. But if the fertilization occurs with a sperm cell carrying the Y chromosome, the resulting zygote will be XXY. That would have been your answer. Now, this was a new way of asking questions. I marked the November 2024, 2025 last year. And I can tell you, in that 11 days of marking, there were only 3 kids that got this right. Only 3. Now, the rest, there was no application, and it was confusing because they were not just thinking about trisomy 21. That's not the question. That's not what the question is about. It was specific. It is happening in the gonosomes. So this was very tricky. I will send you guys the memo now so that you can see everything. Okay, are there any questions on that? Let's do this one. I'm going to send it to the group now. Remember, application application, application, application. So you read into what you are given. Please try your best to answer. That's the diagram. I'm going to show you the questions just now. This is from November 2023. It's probably way too small. Please tell me if it's too small on your side so that I can zoom in a little bit. But I've sent the pictures also on the group.
Spindle fiber for phase one.
Are we done? Or you need a little bit of time? 2 more, 1 more minute?
2 more minutes, please, ma'am.
Well done.
Okay, we'll just give those that are finishing up 2 more minutes and then we start marking. Please, I really need you to try your best with the last question, that non-disjunction question. Please use the chromosome numbers that you find in the cells there and try your best to get that 3 marks. Please, please, please. Okay, let's mark. Let me get my question here. All right, this was from November 2023, I think. So this is quite easy, the first few questions. Give the name of structure B. What did you say, guys, for B?
Centriole.
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 Thank you. Centriole. If you said centrosome, that's also correct. Centrosome. Then for C, what did you guys say for C?
Spindle fiber.
Spindle fiber.
Right.
Spindle fiber. Spindle. My pen is acting up. Fiber. Then identify the phase represented by diagram 1. What phase is that? What did you guys say?
Prophase 1.
Prophase 1.
Thank you. That is correct. Prophase 1. Perfect. If you don't have the number 1, you just wrote prophase. Wrong. You won't get that. Give 3 reasons why you are saying diagram 1 is prophase 1. Please give me 3 reasons. Who's going to speak? Is it Zanele? I see your mic is on. Yes, I'm listening, Zanele. What did you say, Zanele? Now you've just muted your mic. Okay, anyone, please do answer. What did you guys say here? I'm not giving away the answer. I need to listen to your answer first.
Okay, ma'am, I'm
Putlelelo.
not confident about my third point, but like I can tell you my first 2 points.
Give me your first 2 points, Lilo.
So I said in label A, basically where the arrow was pointing down, the inner chromatids from each homologous chromosome overlap and touch each other at the chiasma and at the process that is called crossing over. And then at the second point that I said is that crossing over takes place. Oh, wait, no, sorry. I rewrote that twice. I don't know why. The centrosome splits and the 2 centrioles move apart to form the spindle fibers. That was my second point.
Okay, let's start over. I just want to confirm. Which question are you answering? 2.1.3.
Yes, 2.1.3. Because
Okay.
we were trying to prove that why are we saying that diagram 1 is prophase 1? So I was basically describing the things that I could see within the diagram.
Okay, I'm listening. So what's the first thing?
So what I said at label A, the inner chromatids from each homologous chromosome overlap and touch each other at the chiasma with the process called crossing over.
1 mark for that.
And then the second point I said the centrosome splits and forms 2 centrioles that move apart to form the spindle fibers.
The centrosome splits and
forms 2 centrioles that move apart to opposite poles and form spindle fibers.
Okay, I'll give you that.
And then the third one, I don't feel really like confident on it. Like I didn't even like answer it properly. Hence, I was like, I'm not happy with my third point, ma'am.
Okay, tell me your third point.
I said the chromatid network, has individual chromosomes and pairs of homologous chromosomes that lie next to each other to form bivalent pairs.
You'd get a mark there at the end, but we might penalize you with what you are starting with. So from the 3 answers that you've given me, there's certain parts that are unnecessary and are incorrect, and then there's certain parts that are correct. Now, it will then depend if you find a nice teacher or not so nice teacher. So a nice teacher will say, okay, the first part is unnecessary, but at least there's something correct. So you spoke about you said inner chromosomes. That's what you said, right? First point. Now, there's no inner or outer chromosomes. There's adjacent chromatids, right?
Okay.
So if we are speaking about these chromosomes here, but I understand what you meant. That's what I'm saying. The last point at the end, you spoke about crossing over. That's all we needed. So if this is one chromosome and this is another chromosome, the ones that are going to cross over is these 2, and we say these are adjacent chromatids. We don't call them inner chromatids or inner chromosomes. So one might penalize you for that. But the 3 answers here, we can see crossing over is taking place. That's 1 mark. Don't give us a background information or the point of blah, 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 blah. Crossing over is taking place. We can see it here. We can see it here. We can see it here. We can see it here, or even there. So that is point number 1. Point number 2, I'm trying to remember your points now, but you mentioned something about bivalent chromosomes. So 1 crossing over, that's a point. 2, you are seeing homologous pairs of chromosomes. That's another point, because these chromosomes are in homologous pairs. That's what we can see. Your other point was about the centrosome splitting, forming centrioles. That is also correct, but how we want you to phrase it is centrioles are moving to opposite poles, forming spindle fibers. There is a centriole and there is another centriole. So I would still give you a mark for that, but just the first few words, if you mess them up, that will actually make your whole sentence incorrect. Then another thing we can speak about, what do we call this, guys? This part over here, these dots. What do we refer to these dots as?
It's our
Anyone?
nucleolus. Isn't it our nucleolus, ma'am?
Nucleolus, no. Nucleolus would be
The
inside here.
nuclear, it is the nuclear membrane.
Nuclear membrane. What is
Nuclear
happening to the
membrane.
nuclear membrane?
It's
It
disappearing, ma'am.
disappears.
Thank you so
Disappears,
much.
ma'am.
That's a point.
Okay.
So that's what you see. Nuclear membrane is disappearing. You speak about it. Centriole lines
Okay.
are moving to opposite poles, forming spindle fibers. You speak about it. There is crossing over taking place. You speak about it. You can see the homologous pairs of chromosomes. They are visible. You speak about it. That's a point. Easy peasy stuff. Okay, I'm happy. Describe for 3 marks the process taking place at A. We know the process taking place at A is crossing over. Now describe the process for us, 3 points. What's the first thing? The chromosomes are going to lie next to each other. Chromosomes of the homologous pair will lie next to each other. That's point number 1. Point number 2, adjacent chromatids are going to overlap at a point called chiasma or chiasmata. That is point number 2. And genetic material will be exchanged. That is point number 3. So I'm going to repeat that. Chromosomes or homologous chromosome pair will lie next to each other. That's the first point. And then the second point, adjacent chromatids. These chromatids, adjacent chromatids are going to overlap at a point called chiasma or chiasmata, and then genetic material will be exchanged. 3 marks. That's it. 215. Identify the phase represented in diagram 2. What phase is it? Anyone?
Metaphase 1.
Perfect. Metaphase 1. Perfecto. 215 for 2 marks. Describe the difference. Describe the difference in the events that take place in the phase mentioned in A, meaning the events that take place in metaphase 1, because that's what you would have mentioned, right? And the same phase during mitosis. What phase is that? That is metaphase. So you are differentiating for us. Describe the difference in the events. What exactly are you focusing on? The events that take place in the phase that you've mentioned above, meaning metaphase 1, and the same phase during mitosis, which is metaphase. Tell me what's different there. Anyone? For 2 marks.
Something with the equator.
I'm listening. Is
Ma'am,
it?
ma'am,
Metaphase 1. Okay.
Ma'am, I said that in meiosis crossing over occurs and in mitosis crossing over does not occur. And I said it occurs in meiosis, it occurs in sex organs, and in mitosis it occurs in body cells. Those are the 2 differences that I stated.
Okay, I will note that. I'm noting it. Anyone else with a different explanation? I think it was Zanele and Khutlelelo, your mics were on. Please do give me your answers.
I'm not sure, but I think it's chromosomes line up in the paired chromosomes line up in the equator with metaphase 1. I'm not sure. Then in meiosis, meiosis, I think it's whereby the chromosomes are. Ish, I'm not sure. Someone can help me there.
Okay, I want to write down the answers. The first answer, it was from you, Lilo, and I'm trying to remember everything you spoke about. Just repeat your answer, Lilo, again.
Ma'am, I mentioned that in mitosis, crossing over does not occur, and then in meiosis, crossing over occurs. And then the second point that I mentioned is that in mitosis, it occurs in body cells, and then in meiosis, it occurs in sex organs.
Okay, take a note. Then, Khutlelelo, you spoke about metaphase 1, and you said chromosomes are in pairs. I need your point about metaphase.
I said chromosomes are arranged at the equator, but I think it's where it's singly or they are non-replicated, something like that. I'm not sure.
Okay, and replicated. Anyone else with a different answer? I'm taking 3 options. Anyone else with a different answer? Is it Nambuso? Your hand is up. Okay, let's break it down. So we need to understand the question and we read it to understand it all the time. Now, I always say this to my learners. In an exam, you have 2.5 hours. That is more than enough time to read each question twice before you answer it. So what have they done here? They have asked us to first identify diagram 2. And me and you have successfully done that. And we said it is metaphase 1. They don't leave it at that. They then ask you, describe the events. I want us to focus on the word events. Describe the difference in the events that take place where and where. They are looking at 2 things. The phase that you've mentioned, which is metaphase 1 of meiosis, and the same phase in mitosis, which is metaphase. So our answer about crossing over would be incorrect because crossing over takes place in prophase. Now we are asked about metaphase. So if we are asked about prophase 1 and prophase of mitosis, we would speak about crossing over. If we're asked about general differences between metaphase and meiosis in terms of location, in which specific cells do they occur in, then we can speak about body cells. But the questions have a way of directing you on what exactly you need to focus on. Now, it wants us to speak about metaphase 1 of meiosis and metaphase of mitosis, M for middle. What we then get here is that in both metaphase 1 and metaphase of mitosis, we have chromosomes in the middle of the equator, but there's key words that we need to focus on. Khutlelelo was mentioning them. So when you speak about metaphase 1, you need to speak about the chromosomes arranging themselves in pairs in the middle of the equator. So in metaphase 1, this would be the arrangement of chromosomes. This would be a chromosome and another chromosome attached to the spindle fiber. There shouldn't be in space here. Attached to the spindle fiber. This is a pair. But in metaphase, it would be chromosomes arranged singly in the middle of the equator. That's your 2 marks. So you do mention in metaphase 1, chromosomes arrange themselves in pairs in the middle of the equator. This is a pair. And then in metaphase of mitosis, the chromosomes arrange themselves singly in the middle of the equator. 2 marks. So let's focus on exactly what they are asking. The content is there. I'm happy that you guys are able to give me the correct answers in terms of the content, but we need to focus on the specifics of the question. That's what I need. Okay? Did you get that, Lilo?
Yes, ma'am. Yes, ma'am.
Thank you so much. All right, our last question. This one, I wish I had enough space to write down your answers so I can basically show you how I would mark your answer. So I think the nice thing about saying your answer, you are able to get a response on it, because sometimes you will feel like your answer is correct only to find out that it's not correct. So if my screen comes on and off, Just know I'm erasing. Okay. Now I need to hear your answers and I need different people, please. Please don't be scared. We don't see each other here. We're just hearing people's voices. That's it. Please be confident enough to give me your answer. Samuel, please do give me your answer for this one. I haven't heard you in this lesson. Describe the results at the end of meiosis. If the chromosomes at D failed to separate. Let's go up and see where is D. If the chromosomes at D failed to separate, I need you to apply your non-disjunction content information to answer this for 3 marks. What did you say, someone? Okay, when I call people's names, they don't answer. Nopolisa, Kele, what did you say? I think Evie will also try to answer sometime, and I'm not sure what happened. Let's see. Atli, please also answer. Can someone answer that? Because people are not saying anything when I call out their names. Can someone give me what they've attempted to say here? I'm listening, guys. Okay, maybe let me lead the answer, right? Describe the results at the end of meiosis. Let me start there. Tell me, how many cells are we going to get at the end of meiosis? How many cells?
4.
Well, okay, thank you. I was about to check if people are still here. So how many cells do we get? 4 cells, right
4.
at the end. 4
Mm.
cells will be formed. Because what are they asking us here? Describe the results at the end of meiosis. At the end of meiosis, we get 4 cells. But now, from these 4 cells, let's look at the chromosome numbers. Now, how many chromosomes do we have in the parent cell? In our parent cell, how many chromosomes did we have? We can be guided by this diagram, actually. How many chromosomes did we have in the parent cell? The total number.
Mm.
I'm listening.
Isn't it 5?
Not 5. Let's count from diagram 1. You can count from diagram 1 or diagram 2. How many?
We have 8.
8 chromosomes.
8.
So what are we expecting? Me and you are expecting to have half the number of chromosomes in the cells, in each cell. We are expecting each cell to have 4 chromosomes each.
Oh,
But what has happened here? So if there is non-disjunction at D, so we are expecting a cell to be separated over here so that we can have these 4 chromosomes forming one cell and these chromosomes forming another cell. That's what we are expecting. But now they are telling us, I'm going to erase so my screen will come on and off. What are they telling us now? They are telling us that at D, the chromosomes fail to separate. They don't tell us whether they fail to go up or down, but I guess it doesn't matter in this case. So that means if we were to say the whole pair goes up. So that means this would be one cell, and then I'm going to change my color. And then these chromosomes will form another cell. Tell me, how many chromosomes are we going to find in this cell, this one, that I've represented with an arrow? How many chromosomes? You can easily count the chromosomes.
4 chromosomes.
How many? 5 chromosomes.
5.
5 chromosomes, ma'am.
Or if they fail. It's
Yes,
5.
if they fail to separate.
They
Then
fail.
these ones in blue, how many chromosomes will that cell have?
3 chromosomes.
3,
3
ma'am.
chromosomes. 3 chromosomes. Now, what do we expect to see at the end of meiosis? Let's answer our question. From the 4 cells, that means 2 cells are going to have how many chromosomes each? How many chromosomes each in the 2 cells? 5 chromosomes. So
5
from the
chromosomes.
Correct. From the 4 cells, 2 of those cells are going to have 5 chromosomes each. And what will happen to the other 2 cells? How many chromosomes will they have in each cell?
They'll have 3, ma'am.
Thank you. 3 chromosomes. This is an application question. You use the chromosome number that you are given. We are saying there is non-disjunction here. And this non-disjunction is happening. Yes? This non-disjunction is happening in anaphase 1. Because why are we saying anaphase 1? Because we are given metaphase 1 and these chromosomes are still in pairs. So it will be the whole pair moving up or the whole pair moving down. Now you have to apply the number of chromosomes that you are given. So if now what we had was a parent cell having 8 chromosomes, this is what we're supposed to get in terms of the chromosome number of the cells. But due to non-disjunction, 2 cells are going to have extra chromosomes and the other 2 cells are going to be having 1 chromosome short. So that's exactly what we've explained here. So that's an application question. Any questions on this? Now, this is real application questions. So that is very important for us to understand. That is super, super, super important for us to understand, please. Okay. I don't have time to do the rest of the questions. So I will send you the questions on the group and also the memo on the group. But we can do this one. Let's do this one. This one is 2 minutes, guys. This one is 2 minutes. That's all I can give you. One minute. Okay, let's answer. Let's answer, guys. This one is simple. Just to say bye. This is us to say bye. How many of the following are present in the karyotype? How many chromosomes are present here? Tell me, guys. So at least I can, I drink my medication and sleep. How many?
46.
46.
23.
46.
23. Am I hearing a 23? I heard a 23. There is someone that said 23. If you just wrote 23, that would be wrong. But if you said 23
I wrote
pairs,
20.
you wrote 20.
Yeah.
I said 46, Miss.
Or 40.
23 pairs.
Okay, 46 is correct. 23 pairs is correct. I'm hearing someone saying 20. 20 will be
Yes.
definitely in. How did you get 20? Is it Mulalo? How did you get 20?
I get 20 because of I read it like this. The screen is gone now. Okay, I read it like this. Okay, it was says it says chromosomes, and then B says autosome, and says chromosome. So I was reading like, okay, I have to find gonozome, I have to find autosome, I have to find chromosome. So I didn't include all of them. That's why I end up with this 20.
Okay, so these are separate questions. It's just a matter of checking if we understand the terminology here. So with the chromosomes, we are physically counting them. 1, 2, 3, 4, you should get to 46. Okay, then autosomes, how many autosomes did you guys get? How many?
44.
44.
22 pairs, ma'am.
22 pairs is correct. Check where the tick is at, it's after pairs. So if you just said 22, no mark. Then gonosomes, how many gonosomes do we have? Here, what you can see on the diagram. How many gonosomes?
2 pairs, ma'am. So basically 4.
Can we see 4?
Wait, ma'am. Let me open my eyes.
Wide open, please. These are the gonosomes.
Yes, it's number 23, ma'am. So there's 1 pair. My bad. I'm sorry, ma'am.
So, ma'am, I wrote one. I wrote one. Is that correct?
Hi.
Or one pair?
You guys always make my day. You make
So
my
we
day.
have to. So
Sorry,
we
Yes,
have
ma'am. It's
to
been a
I'm
write.
long day,
listening.
ma'am.
So we have to write 2 and or one pair.
You just have to choose which answer you like. Both are correct.
Okay,
So
I wrote one pair. Is that correct?
Mulalo, did you write just one or one pair?
I wrote one pair,
Okay, one
not
pair
2.
is correct. If you wrote just one, wrong.
Yeah, I wrote
Okay,
one pair.
okay, that is correct. Okay. How many chromosomes would be present in the gametes produced by this individual? How many chromosomes? I don't want to get
23.
more sick. I'm hearing an answer. 20?
23, ma'am.
Perfect. 23, because they're speaking about what? A gamete. A gamete will have half the number of chromosomes. Yes, I'm hearing someone. Okay.
So, ma'am, ma'am, if I just wrote 23 is correct without including anything, right?
For 1.4.2, yes.
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 Okay.
Because they're
If I
asking
wrote
about
1
the
point.
gametes. If you write pairs here, then that's wrong. Because we are saying gametes have 46 chromosomes. So in terms of gametes, there are 23 chromosomes, and that's where it ends.
Okay.
Okay. Last question. Is the karyotype in the diagram that of a female or male?
Male.
It's a male, ma'am.
Male. Male. Give me a reason. Why are you
It's a
saying it's
male,
male?
ma'am.
Give me a reason, please.
It's because there's the Y on the gamete number
There's an X
23.
and Y.
It's an X and Y. The gonosomes are X and Y.
And Y.
All right. I think I'm feeling a little bit better, but now let me see if I can project another one that is quick. Do this one, guys. Do this one. At least now I have my water and lemon. 5 minutes.
Still terminology.
Yes.
Okay.
Can we mark? Yes. 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 No.
No, man.
Not yet.
Yes.
Okay, let's do it quickly. Number one, what did you guys get? The division of the cytoplasm after a cell nucleus is divided. What did you guys get?
Cytognosis.
Cytokinesis or cytokinesis. Okay, number two, what did you get?
Phyto-
Chiasma.
Chiasma. Chiasma.
Chiasma or chiasmata. Then number three,
Non-disjunction.
Non-disjunction.
yeah,
And a
yeah,
phase.
yeah.
Non-disjunction. Phase one.
Okay, my pen is disappointing me now. Then number 4.
Centriole.
Hmm?
Centromere.
Centromere.
Number 4, it's centromere.
Thank you. 5.
Number
There you go.
Hey,
Did we get that?
okay.
No,
No.
No.
We didn't get this
no,
one.
miss. Yes.
Oh.
So remember, we have haploid and diploid. If you're only having a single set of a pair, that's haploid. If you're having a full set, that's a diploid. 6. What did you guys write in 6?
Spindle fiber.
Thank you. Now the fiber can be written as BRE or BER. Both are accepted. 7. What did you guys write?
Network.
Why are we saying it's a chromatin network? Remember, it's a network. Then when the cell is about to divide, if a cell is non-dividing, it's chromatin network. Then it condenses to form chromosomes when the cell is about to divide. So that's why we're having it as chromatin network. Number 8. Number
It's
8
chromosome.
is chromosome. If you wrote replicated chromosome, correct. Number 9. What did you guys say? Number 9.
Interphase.
Thank you. That's correct. Then number 10. I'm very interested in this one. What did you guys say? I'm hearing the laughs.
Random arrangement.
Random arrangement of chromosomes. That's what we have. I didn't think I was going to be able to do 2 hours, but here we are. I'm going to exit from here. I need you to tell me how you want us to move forward with this. For next week's lesson, do you want me to send you some activities of meiosis? Remember, there was a topic test for meiosis that I sent. So I'll send a memo for that. In next week's lesson, do you want us to continue with meiosis, do different questions or mark that topic test? Or should I start genetics? So tell me what you would prefer.
Genetics.
Please,
Let's
start
start
genetics.
genetics.
Please start genetics, miss.
Okay.
And please send us questions so that we can answer during the week.
For meiosis.
Yes.
Okay, I will do that. So meiosis questions and memos this week. And then we start genetics. It's going to drag a little bit because genetics is long. So I'm happy that you guys are ready to start genetics. But let this not be you closing the chapter with meiosis. I'm sure you are picking up mistakes. Use this week to finalize and close any gap that you have when it comes to meiosis. It is one of the topics that is failed in paper 2 because of terminology and because they are very strict in how they want you to answer. So please, please, please let this not be the last day you read or study or do a meiosis question. I will sort you out with the memo for that meiosis topic test and with some questions maybe tomorrow, latest on Thursday, but I should be fine with sourcing some questions and sending them to you guys tomorrow. Please, let's not be too quiet on the group. So just if you're answering, just ask a question. Is this correct? Is this not correct? or something so that we become interactive and you get all the help now. Because a day or a week before your exam, I don't want 100 questions on that group. I won't be able to answer all of them. So let's just maximize our time now. All right.
I'm
Next
sorry,
week.
ma'am. Before you conclude, I have a little question that I would like to ask you, ma'am.
Yes.
So, ma'am, I was